Hello and welcome everybody to my channel ominibioentrance, and today we will be seeing CSIR PYQs of June 2023 Shift 1. I will be dealing with applied biology and methodology, DNA replication, protein synthesis and genetics. Then next one. The following statements are made about the digestion of proteins and enzymes involved. Which of the following options represent the combination of all correct statements? Chymotrypsin does not generate peptides with C-terminal neutral amino acids. Chymotrypsin cleaves C-terminal of phenylalanine, tryptophan, tyrosine, etc. and all three are neutral amino acids, so option A is not correct. Trypsin generates peptides with C-terminal basic amino acid, that is correct. Carboxypeptidase B acts on aromatic amino acid. It acts on basic amino acid lysine and arginine on their C-terminal side. So C is not correct. Carboxypeptidase A employs zinc ion for hydrolysis. That is correct. The brush border enterokinase has no polysaccharides attached to it. That is incorrect. The final digestion of two amino acids occur in the intestinal lumen, the brush border and the cytoplasm of the mucosal cells. That is correct. So B, D and F are correct. So the answer is option 4. Coming to the next question. The following statements were made about phase contrast microscopy. Phase contrast microscopy can be equally utilized to examine stained and unstained specimens. It is usually used for unstained cells, not for stained ones. So option A statement is not correct. A phase annulus generates hollow cone of light to illuminate the specimen. That is correct. A light wave that passes through the cell nucleus and organelle lags compared to the light wave that passes through water only. That is also correct. A polarized light source is used to translate the minor phase shifts into gray values. It is not polarized light source, but a phase plate is used. So D is not correct. Appearance of bright halos is a common artifact of phase contrast imaging. That is also correct. So B, C, E is correct. A and D are wrong. The next one. The LAC-I and TRPR repressors bind to their ligands allolactose and tryptophan respectively, resulting in alteration of their DNA binding properties. The following statements are made about the mechanism of LAC-I and TRPR binding with DNA and regulation of gene expression in E. coli. So first option statement A. Allolactose binding to LAC-I lead to its poor binding to the LAC operator, whereas tryptophan binding to the TRP leads to its better binding to the TRP operator. That is correct. Allolactose binding to LAC-I lead to induction of LAC operon. That's correct. Whereas tryptophan binding lead to repression of the tryptophan operon. That is also correct. Binding of allolactose and tryptophan to LAC-I and TRPR respectively lead to repression. No. B statement is correct. C is not correct. Binding of allolactose and tryptophan to LAC-I and TRP respectively lead to activation of their corresponding operons. That is also not correct. It is not activation. One is induction and the other is repression. The answer is option 1, A and B only. Then given below are few terms related to bioinformatic resources, column X, and their functions or applications in column Y. Which of the following options represent all correct matches between column X and column Y? Tremble, TREMBL. TREMBL is database of protein sequences. TBLAST-N. TBLAST-N is comparison of amino acid sequence against nucleotide sequence database translated in all 6 reading frame. SCOP. SCOP is manually curated structural classification of proteins. Join map. Join map is analysis of recombination frequencies between molecular markers. So the answer here is option 1. A2, B3, C4 and D1. Coming to the next question. In 4 taxa A, B, C, D, 2 characters, shape and color were scored to infer their phylogenetic relationship. The 2 character states for shape were square and round, while the 2 characters for the states color were black and yellow. The character distribution is given in the table below. Using the above data, 4 trees were built using the method of maximum parsimony, which are given below. Select the option that represents the most parsimonious tree. Now, what is parsimonious tree? It is a tree with fewest number of evolutionary changes. It will be the most parsimonious tree. So, out of these trees given here, which is the most parsimonious? So if you see A, you can see it is black in color, first a square, then square but yellow in color, then it is coming to circle. So 2 evolutionary changes have happened. That is change in shape over here from square to circle and change in color from black to yellow. So 2 changes. If you see B, there is change in shape. First case, then change in color, then again change in shape. So totally 3 changes. First change in shape, then change in color and change in shape again. So it is 3 changes. So B is with 3 changes. C we will see it is change in shape as well as color. Then again change in shape, then again change in shape. So totally 4 changes. So C is with 4 changes. D, if you see, there is change in color, then there is change in shape. So only 2 changes. So out of these, the one with fewest number of evolutionary changes will be the most parsimonious. So if you see this, you can see that A and D are having only 2 changes each, and so they will be the most parsimonious. So answer is option 4, A and D. Next question. Assume that the genes W plus and CV plus are located 10 centimorgan apart on the X chromosome of Drosophila melanogaster. Mutations in W plus and CV plus give rise to white eyes and cross-veinless phenotypes respectively, which are recessive to the wild type phenotype. A homozygous wild type female was crossed to a white-eyed cross-veinless male. The F1 progeny was sib-mated. Sib-mated means selfed. What percentage of the progeny will be white-eyed and cross-veinless? So this is the female genotype X. They have told it is wild type female and cross-veinless and white-eyed male. So W plus C plus will be the female and male will be XWC and Y gametes. So the gamete for the female will be XW plus C plus and male will produce 2 gametes. Upon mating, you will get the F1 progeny as XW plus C plus XWC and XW plus C plus Y. Now F1 is sib-mated. These are the gamete parents, which are one is male and the other is female. These two are self together, and these are the now this one, the female will produce parental gametes as well as recombinant ones. They have told that the two characters are 20 centimorgan apart. So the recombinance will be 20%. That is, this gamete will be 10% and this gamete will be 10%. The rest, the parental will be 100 minus 20, that is 80% will be parents. So 40% will be this gamete and 40% will be this gamete. Now the male will produce only two type of gametes, so 50%, 50%. Now you have to draw the square, Punnett square, and see. So this is the Punnett square. These are the gametes. 50%, 50% will be male. 40%, 40% parental gametes. 10%, 10% will be your recombinants. So they have asked which one will be white-eyed and cross-veinless. So this is the only progeny that is white-eyed and cross-veinless. That is 40% into 50% into 100. That will give you 20%. So the answer is 20.